Paul Bolno, CEO of Wave Life Sciences. I'm here with Anne-Marie Li-Kwai-Cheung, our Chief Development Officer. Um, and we, again, work at Wave Life Sciences, where we are focused on translating RNA medicines for patients with a variety of genetic disorders. I think we're still very much at the beginning. I think we've learned a lot about and it's not just the market. I think we've learned a lot about the disease and various approaches to being able to treat that. So I think for the vast majority of the beginning, it was really solving for inflammation. So the history of bringing steroids and treatments to treat muscle inflammation forward to slow the progression of the disease. I think what we've seen a lot of over the last several years, which is, I think, exciting for the community, is really a focus on ways to restoring dystrophin protein. So how do we not just prevent the acceleration of the disease by slowing inflammation, but how do we actually think about getting to the underlying challenge, which is protein restoration? How do we fix the protein to prevent it from declining? And therefore, as a starting point, how do you prevent the continued acceleration of the disease? But important for the future is how can one think about improving it and regressing it? I think we also have to step back and realize that we talk and discuss a lot in therapeutics about the community as being singular. And I think if we think about the journey in Duchenne muscular dystrophy, there really are distinct components. There's a lot of discussion right now in the therapeutic landscape about ambulatory boys. And these are boys of various ages where they still have the ability to walk. However, that continued increase in that muscle loss continues to put those boys at risk of losing ambulation. I think there's another community that's extraordinarily important, which is the loss of ambulation, where therapies that restore dystrophin are still very, very important, meaning how can you protect respiratory function by protecting diaphragm and lung and cardiac? How do you prevent the heart from going into cardiac failure? So the muscles wasting within the heart. So a very important opportunity still for treatments that affect the later stage of the disease after loss of ambulation. I think another area that's emerging, and I think this is with the recognition that the community's done a wonderful job of bringing newborn screening in, both in Europe and in the US, with the realization that oftentimes patients have a multi-year journey to the diagnosis of Duchenne muscular dystrophy. And at that time, and in this community, that they know well that time is muscle, that the delay in finding out that you have Duchenne muscular dystrophy is a loss of getting on therapy. And so with newborn screening, there's this emerging area, which is how do you treat young children before that stage of loss of muscle? So if we think about the area where our study was looking, where a lot of studies are able to look, it's in ambulatory boys, and we're studying that piece in the middle. But the opportunity for our therapeutic, and I think others that restore dystrophin, are really to be able to look holistically at the entire Duchenne muscular dystrophy community. So if we think about the therapies of exon skipping strategies, they're really designed to think about the fact that you've got this very, very big transcript, and you can have a variety of deletions in that transcript, meaning areas where there's a piece missing, not in frame, and you're not making a protein. And the concept of exon skipping is really to shift that transcript back into frame, kind of by putting almost like I want to say a Band-Aid is a natural way of trying to describe it over an area of deletions that pull a big three-dimensional protein back into a different configuration, pull it back into what's called in frame. And by doing that, then the machinery inside the cell can start producing isoforms of dystrophin, meaning you can produce various lengths of different dystrophin protein. And this whole theory and thesis is really predicated that there is a community, the Becker muscular dystrophy community, that have a variety of these smaller truncated isoforms of various sizes that allow the protein to retain its functional activity, meaning the patients don't lose the ability to lose ambulation. And so the theory in exon skipping is if you could restore these to various dystrophin isoforms and they can be very small, then you can ultimately have the ability to fix what's missing inside the body. And there's a lot of resiliency in that dystrophin protein to reduce its size. It's a remarkable protein in that effect, that you can really make this protein small and it can still retain its effect so long as it has core elements. And those are the elements that are evaluated as part of the dystrophin analysis. So if you can do that and restore that phenotype, that's really the basis of saying, as I was saying earlier, if we can bring dystrophin back, and I say there, dystrophin in the big context, then you have that ability and to fix muscle, hopefully prevent loss of ambulation and see benefits in the physical metrics. But I think what's unique, we've looked at it and evaluated it preclinically, and I think it's one of the unique aspects of N531. We've shown preclinically that we can get that medicine, that N531, preferentially into heart and diaphragm. We actually see higher levels of muscle concentration in the heart and diaphragm over skeletal muscle. Meaning, as I was saying earlier, if we can get this distributed to the broad muscle cells in the body, you do have that potential to look forward to seeing improvements in other areas. It just takes time.
FORWARD-53 is a phase 2 study in boys with Exon 53 amenable DMD. It's 48 weeks in duration, and we have recruited 11 boys into the study, 10 of whom are ambulatory. It means they're still able to walk, and one boy who is non-ambulatory, which means the stage of his disease has progressed to the extent he can currently not ambulate reliably. We recruited patients with a mean age of 8 years old. The study has just reached its 24-week interim time point. The study is looking at the safety and tolerability, pharmacokinetics, and pharmacodynamics of N531, with the primary endpoint being dystrophin production. We're looking to see that N531 results in a change from where the boys would have started, which would have been having no dystrophin, to the midpoint and the end of the study. And we're also collecting functional endpoints that are used to measure how the boys are generally performing, as well as a digital endpoint. This is called 95% stride velocity, and this looks at essentially the speed and strength of the steps that the boys are taking. It's a remote sensor that the boys can wear at home and is collecting data passively so that we can see how they're doing and assess the impact of the drug on their ability to walk. At the 24-week interim endpoint, we're really delighted to announce that we have seen a 9% muscle adjusted dystrophin response, which is 5.5% non-adjusted. And I think really importantly, across the boys in the study, we saw a really consistent response with 89% of ambulatory patients reaching more than 5% of dystrophin. And the reason that's important is because there have been other therapies on the market that have reached a mean of more than 5%. And the 5% is relevant because this represents a lower bound for another type of dystrophy called Becker muscular dystrophy. Those patients have much later loss of ambulation and a longer life expectancy, and they produce isoforms of dystrophin. And patients who have at least 5% have a much milder phenotype. So 5% is a really key level for everybody to remember. And in our study, really 89% of the ambulatory patients have reached that 5%. So this means that these patients are really getting the best opportunity to experience some benefit in future of having restored dystrophin levels.
I think Anne-Marie's point is a really important one when we think about the landscape of medicines for Duchenne muscular dystrophy. And I think that's this idea of consistency. I think for a long time, in a very important way, we talk about quantity, how much dystrophin is being restored, that context of if you can skip that protein, you can make these dystrophin, isoforms that create protein. That's important. But I think what people forget is it's not just about quantity. The Becker phenotype, this Becker muscular dystrophy that we refer to that has this improvement, these people have dystrophin expressed throughout the body, and it's consistently across the patients that are in these outcome studies. And so we set this bar and see that 5%, you see a really meaningful difference. One has to step back and realize that that data was analyzed around a number of people that all have that same consistent expression of greater than 5% dystrophin. And I think in clinical trials a lot now, we tend to look at a mean number, and everybody focuses on a mean number as if that mean number is equivalent. And I think really stepping back, and I think it was an important part of the data set that we shared, was really the patient level data. How is that 5% or more distributed across the patients who are on the study. And I think that's going to be important as the field moves forward to really examine and explore when we say X% dystrophin as a mean, understanding how patient responses are happening are going to be important thinking forward to say, when you run larger studies, when you continue the study to look for potential benefits, making sure that you've got the vast majority of patients on those studies who are actually experiencing that greater than 5%, which is a threshold.
So we've reached the 24-week interim analysis, and the study is 48 weeks long. So the FORWARD-53 study is continuing. In addition to that, we are going to be engaging regulators on the pathway to accelerated approval. And this is all expected to conclude in Q1 2025. So, we're close to finishing the study. At the end of the study, it will also be a great opportunity for us to analyze all of the data, including the functional outcomes and the digital biomarker that we have been collecting, and have another update on the safety and tolerability, which was actually really good at the 24-week time point. There were no serious adverse events. Treatment-related adverse events were all mild, there were no discontinuations. So we'll certainly be looking at the safety as well as the PK and PD at the 48-week time point. We're excited to finish the study and take N531 to its next steps.
There really are some big differences beyond dystrophin, because I think people hit dystrophin as kind of the answer, but we saw not just high dystrophin and consistent dystrophin, but also we got into areas of the cells that nobody else is getting into. So stem cells that are in this regenerative repair, we show for the first time you could get distributed into the repair cells, which helps think about treating earlier. In addition to that, heart and diaphragm. So I just think there are some points of differentiation where we're seeing a real different profile beyond just the magnitude of protein expression.